Hello, my name is Dr. Gaynor Smith, and I'm delighted to be asked to be part of the Neurodegeneration Institute webinar series. I was previously employed in the Ole Isaacson lab at the McLean Hospital, Harvard Medical School, and it's there that I really found my passion to study mitochondria in the context of neurodegenerative diseases. After my first postdoc, I then went on to the University of Massachusetts to complete a second postdoc fellowship under the supervision of Professor Mark Freeman, who then moved on to Oregon Health and Sciences University. And it's there that I started to study the humble fruit fly, or Drosophila melanogaster. And now I am a new faculty at Cardiff University, UK. And there I've combined both these passions, and now I study mitochondria in the fruit fly. And I'm delighted today to present some of my work concerning an unbiased genetic screen to look for new modifiers of mitochondrial biology in the neurons of fruit flies. So I will now share my screen with you and talk you through some of my work. So I'm going to talk to you today about the genetics of mitochondria, specifically in the long axonal projections of neurons. So what we do two things within the UK Dementia Research Institute, or DRI. And we use Drosophila to both model human diseases that have a genetic identity. And we also perform unbiased genetic screens using our fruit flies to gather information for further studies, either in cells or in mammals, which could go on to identify new genetic targets for the therapeutic pipeline. So in the lab at the moment, we are using human data sets such as genome-wide association to identify risk genes in Drosophila and model these in terms of causing phenotypes linked with Alzheimer's. I'm also using a similar approach to look at genetic factors which may increase your age of onset of contracting Huntington's disease. I'm interested in how mitochondrial dysfunction contributes to aging, and finding new genes that modify mitochondria, which each could be potentially a new therapeutic target. And it's this last point that I'm going to talk to you today. So why use the fruit fly in neurobiology and neurodegeneration research? Well, they have a small defined brain. We can map out every single neuron within their brain. They have an extremely rapid life cycle. So from embryo to adult flies typically takes just 10 days. And then a fruit fly can actually last to about 60 days, depending on conditions. And we have an array of powerful in vivo genetic tools that we compare with our Drosophila research, which makes them highly genetically tractable. And in my lab, we usually pair this with high resolution clonal imaging technologies. And just like their mammalian counterparts, they have just about every cell that you would expect. So they have supporting glial cells, they have ensheathing glia, which typically wraps neurons, and they have astrocytes, and they have all the different subtypes of neurons, GABAergic, glutamatergic, and so on. What's nice about using Drosophila in research is that we have a number of different depositories where we can buy pre-made fruit flies, if you like, in which we can do a variety of different experimental techniques. So we can buy, for example, flies harboring technology to knock down almost every gene in the Drosophila genome. We can make transgenic flies, and we can make transgenic flies that harbor a human gene. We can do organelles, directed expression of cDNAs. We can tag proteins with fluorescent tags to monitor them under the fluorescent microscope. We have cell type specific transcriptional reporters and biochemical reporters, and a whole library of different mutants. And of course, where mutants don't exist, we have the capacity to make our own using the CRISPR-Cas9 gene editing technology. So this is a beautiful photomicrograph of a mitochondrion. And at this ultrastructural resolution, you can make out the elaborate folds of the crystine and the inner and the outer mitochondrial membrane. And it's on these cristae, that's where the electron transport chain sits. And of course, the primary purpose of the electron transport chain is to generate a proton gradient and ultimately generate energy in the form of ATP. And of course, this is fueled by the redox reactions of the Krebs cycle. And mitochondria are found just about everywhere in the brain cells or the neurons. They're found in the dendrites, in the cell bodies, and down these long axon projections. Now, in order for a mitochondrial to function adequately in the neuron, it has to undergo a number of key processes. So mitochondria have to be born and go through a process of biogenesis. They have to separate from one another through a process called fission. They fuse with one another to share their components called fission, and old damaged mitochondria have to be degraded, and they have to be transported in the cell to where energy is needed. And this is a nice example of why mitochondria are so important in neurons. And this was a study that came from the worm, or C. elegans. So here you can see on panel A, if you were to severely cut an axon of a neuron that contains mitochondria, 24 hours later, this axon stays intact. However, if you cut an axon where a mitochondria is not present, it no longer remains after 24 hours. So they're extremely important, not just for energy generation, but also to buffer these calcium influxes, which ultimately cause neurodegeneration. And this is a beautiful trace of a filled dopaminergic neuron from a rat. And this is a single dopaminergic neuron, and it actually makes about 1 million synaptic connections. And if you were to add up all the arborizations in these axons, it actually reaches over 1 meter in length. So you can imagine that the struggle of neurons like this to maintain mitochondria at the appropriate levels, at the appropriate concentrations needed to adequately supply energy for a cell so enormous, such as the dopaminergic neuron. And of course, mitochondrial dysfunction has been linked to a variety of age-dependent neurodegenerative diseases, such as Parkinson's disease, where you typically see an increase in ROS, or reactive oxygen species. In Huntington's disease, you see a reduced mitochondrial membrane potential. And in ALS, for example, you see increased mitochondrial DNA mutations, particularly in the motor neurons. So it's important to study these organelles at the basic level, so it might one day help with neurodegeneration research. So far, we know the key players, the key molecules, if you like, that regulate mitochondrial neurons. So for example, we know that two molecules called pink and parkin interact with each other to initiate a process called mitophagy, or mitochondrial degradation. We know molecules such as DRP1 and FIS1 are required for mitochondrial fission, and mitofusins and OPA1 are required to fuse to mitochondrial proteins. Other research in Drosophila has discovered two more molecules called Miro and Milton, which are required to remove mitochondria off the cytoskeleton in areas of high calcium. So we know some of the key molecules involved. However, during my time in the Freeman Lab, we devised an unbiased genetic screen to find brand new molecules that govern mitochondrial maintenance in neurons. And we did this using adult Drosophila, which gives us an in vivo system in which to study these energy generating organelles. So just to talk you through the methods of the screen, first of all, so we used a chemical called EMS to induce random mutations into the fly genome. And we were looking at mitochondria in peripheral neurons. So these are long neurons that run from the tip of the wing. And we were able to study mitochondria to where the wing joins the body. And using genetic techniques, we were able to label the neurons in red and the mitochondria they contain in green. And we used a clonal approach called Marf for several reasons. So Marf enables us to look at just a select handful of neurons rather than looking at a whole nerve bundle. And so that we can see mitochondria at the single axon and single mitochondrial resolution using confocal microscopes. Secondly, what Marf enables us to do is to potentially look for homozygous lethal mutations, but since we're only looking at in the neuronal clones, and the rest of the Drosophila is essentially wild type, we're able to bypass this lethality and study even the most severe mutations by looking at them in clones. And using random mutagenesis approach, we were hoping to find modifiers of the key processes that I mentioned earlier. So biogenesis, fission, fusion, transport, or degradation. So this is what these mitochondria look like under the fluorescent microscope. You can see the white arrows are labeling the mitochondria in green, and the neurons themselves are labeled in red. And first of all, we wanted to see whether this screen would work. So we wanted to look at the mitochondrial phenotype that could be seen with known mitochondrial mutations. So if we knock out Milton, or MILT, as it's annotated here, you can see that there are no mitochondria in the axon, and this axon gets degraded away, which is what you would expect because this molecule is involved in transport. If we knock out a mitochondrial fission gene, mitochondria are no longer able to separate apart, and you can see the emergence of very long mitochondria. And conversely, if you knock out a mitochondrial fission protein, so Marf at the bottom, you can see that there are many more smaller mitochondria, which is indicative that mitochondria can no longer fuse, so they stay in the small formation. So we were confident that that screens works, that it was verified, and with that, we wanted to look for random mutations which could cause deficits such as these. So I'm going to talk you through some of the candidates, or as we say in the Drosophila world, hits from the screen. So here you can see two examples where we've hit a gene which causes mitochondria to be balled up within the axon. And you can see each balled up mitochondria is associated with an axonal swelling, which just goes to show you that mitochondria and axon integrity are tightly linked. So we have another mutant where there are less mitochondria in the axons, which may point towards that we've hit a molecule that's involved in mitochondrial transport. In one particular mutant, we see the emergence of very long mitochondria within the axon, even longer than we saw with the DRIP1 mutant. And in others, we see sort of fragmented mitochondria within the axon and cell body. So I'm going to talk to you today about this mutant, which gives us the long axonal mitochondria. So here we've actually quantified both the length and the number of mitochondria within the neurons. So the three panels at the top here are our controls at different ages. So a fly at 2 days in adulthood, 7 days, and 28 days into adulthood. And this is pretty old for a fly. And at the bottom, you can see the same ages in our mutant. And what we find is that mitochondria do increase in size a little as flies age, which is also seen in our mammalian counterparts. But with this mutant, we see the emergence of very long mitochondria from about 7 days. And as we see the occurrence of these long mitochondria, the number of mitochondria is reduced in this axon. So this really is an age-dependent phenomenon. And these size differences can be illustrated when you separate mitochondria into different size bins. So you can see that mitochondria, there are a few mitochondria that are less than 0.2 micrometers and no mitochondria in the control or wild type conditions that are over 1.8 micrometers. However, even at 2 days of adulthood in our flies, we see some mitochondria even longer than 5 micrometers. When we look at later time points, so 7 days, you can see that the frequency distribution of this histogram is shifted, and we see many more longer mitochondria and less shorter ones. So was this a weird phenomena that's just seen in the peripheral neurons? So we actually can dissect out the fly brain from the head and look at the mitochondria in neurons of the central nervous system. And we see long mitochondria in these neurons and sort of hyperfused phenotype in the cell bodies. And we also wanted to look down at the synapses, and this is just in photomicrographs from the neuromuscular junction. And interestingly, even though we see phenotypes in the cell body and the axons, we don't see a mitochondrial phenotype at the synaptic terminal. So this really is interesting, and it points towards cell compartment specificity. which is sort of an untapped field. And it's hoped through unbiased genetic approaches such as this, we are able to determine what governs mitochondrial size and position at different points within the neuron itself. And using genetic tools, we're also able to look at mitochondria in the surrounding glial cells. And what was interesting when we did this was that mitochondria are very long, even in control glia, much longer than you would ever see in a neuron. And when we looked in our mutant, they were very similar to control. So this does seem to be a neuron-specific phenotype. We also decided to look at different subtypes of neurons. So we looked at glutamatergic neurons. We looked at cholinergic and dopaminergic neuron, and this long mitochondrial phenotype manifested in all the different neuronal subtypes that we studied. So this wasn't something specific about the glutamatergic neurons. It seems to have a general effect. So our hit, which we originally called number 541, turned out to be a mutation in a glutathione S transferase gene. And the classic Drosophila experiment to prove definitively that you're looking at the correct gene is to reintroduce your gene into the mutant background. So here you can see our phenotype again, where there are longer mitochondria in the axon and their number is reduced. However, when we reintroduce our gene of interest into the neuron clones, the phenotype is restored, numbers return to normal, and also the length of mitochondria returns to normal. And what we also did that I don't have time to go into today is that we also managed to rescue the phenotype using the human glutathione S transferase homologue of this gene. Which indicates that this may be important in human neurons as well. We were also able to rescue the phenotype by just expressing the glutathione S transferase component of this gene, which further indicates that it's the enzymatic action of this transferase that's actually causing the phenotype. So what do glutathione S transferases do? So glutathione S transferase conjugate glutathione in its reduced form, annotated as GSH here, and it conjugates glutathione to oxidative proteins and lipids to detoxify the cell. So it's important in redox balance. And glutathione itself cycles between its reduced state and its oxidized state through the action of peroxidases and reductases. So how can this glutathione S transferase impact on mitochondrial number and morphology? So first of all, we wanted to see where this protein was localized. So we decided to add a fluorescent, green fluorescent protein to our GFZF cDNA, and we were able to visualize it under the confocal microscope. And as you can see, it has ubiquitous expression within the neurons found in the axon cell bodies and down at the synaptic terminals. So we also wanted to see whether the function of mitochondria was impacted. So we're able to use a combination of genetic tools expressed within our clones, and we combine this with in vivo live cell imaging. So we're able to essentially immobilize the fly on a microscope slide and image these mitochondria live. So you can see that if we look at the calcium levels through analysis of the GCAM fluorescence intensity and compare between our mutant and wild type, there doesn't seem to be a difference in the calcium levels. There wasn't a difference in the Rho GFP, which is an indication of oxidation, but this was a general Rho GFP. And we were also able to look at the age of mitochondria using a tool called Mitotimer, which moves from the fluorescence spectrum shifts from green to red as the mitochondrial age. And there was no differences there either. So this all indicates that mitochondria were broadly functioning as normal, which gives us an indication of why these neurons stay intact. We did go one step further than this, and we looked at very specific redox reporters that could measure levels of either glutathione in its oxidized state or hydrogen peroxide levels. And what we found when we used these more specific reporters was that we do in fact see an increase in oxidation in our glutathione knockout mutant, but no changes in the levels of hydrogen peroxide. So we think about the emergence of long mitochondria through potentially two processes. So long mitochondria could either come about because of enhanced fusion or perhaps decreased fission. So we wanted to test this. We could do this using genetic epistasis experiments. So here we've taken our neuronal clones which have the mutation, and this time we have overexpressed Marf RNAi, which should knock down the molecule Marf. And what we find when we do this is that mitochondria return to their wild type length. So when we knock down Marf, mitochondria become shorter and their number increases, similar to that of control. So what it tells geneticists is that our gene must be upstream of Marf, such that when we knock out our gene, it increases Marf and increases mitochondrial fusion. So this fills in a number of gaps in our jigsaw puzzle. So we now know that when we knock out our Gtzt, it somehow causes an increase in these oxidized glutathione species, which increases mitochondrial fusion through Marf. However, how this occurs remains unknown. So we did some further genetic epistasis experiments to determine this. And this slide is a little detailed, so please don't look at this too heavily. But needless to say that we found out through these tests that the increase in oxidized glutathione happens not because of something passive, but because of this glutathione peroxidase, so GPX here, and not because of any other way to imbalance the system. But we still didn't know really how oxidized glutathione could increase Marf levels. So from here, we turned from our humble Drosophila and we looked at some mouse primary cortical cultures. And we used two small molecules on these cortical cultures. The first one called BSO inhibits the synthesis of reduced glutathione, and the second, called diamide, rapidly converts glutathione in its reduced form to its oxidized form. And we wanted to see the impact of these small molecules on mitochondrial morphology in the axons of these neurons in vitro. So you can see if you use either one of these small molecules, it causes the occurrence of very long mitochondria in axons, which when quantified reduces their number. So we also went on to look at mitofusin 2 levels via Western blotting. And what we found was interesting, that compared to our vehicle treated cells, if we treat these cells with diamide or BSO, it causes an increase in high molecular weight species of mitofusin, but it has no real effect on the monomeric forms. And this is quantified here. So it is ultimately changing glutathione, but only in these large oligomeric forms. We looked at other molecules involved with mitochondrial dynamics, and we, for all intensive purposes, we didn't see any change in the levels of these proteins. So we can conclude that the mitochondrial fission seen both in vivo and in vitro is to do with this high molecular weight species of mitofusin. And we also fixed our primary neurons, and we looked at mitofusin using specific antibody, and we could see enlarged puncta indicating that there are more oligomeric forms with treatment with diamide and BSO. So we were able to show that if you knock out our Gtzt gene, this would lead to an initial increase in glutathione, which is rapidly converted by peroxidases to its oxidized form, which ultimately increases the oligomerization of Marf. And to draw parallels with our Drosophila experiments, we also looked at the mitochondrial physiology with these drug treatments. And here we can see in vehicle conditions, if you use a TMRE, live mitochondrial dye, mitochondrial membrane potential is normal, and you can see that by the red fluorescent intensity. And thiol levels, which gives you an indication of the glutathione redox state, are high, as shown here in the blue. If we use mitochondrial uncapillin protein, a small molecule called FCCP, you can see that mitochondrial membrane potential is lost. And if we use sort of small concentrations of BSO and diamide, where this long mitochondrial phenotype occurs, we can see that actually mitochondrial membrane potential is maintained. So this is similar to our fly experiments, that if you use a low dose of these molecules, it causes fission, but the mitochondria are still able to function. Obviously not the case at higher doses. And at these small doses, we were able to see a decrease in the thiol stain as well, which indicates that glutathione is changing and the free thiol species with them. So we went one step further in our primary neurons, and we decided to use a CRISPR-Cas9 approach to generate clonal knockout cells of the gene that was homologous to our GFZF gene, so Gtzt1, and measure the mitochondria within these knockout axons. And similar to what we observed in Drosophila, we saw the emergence of these long, more clumped up, if you like, mitochondria within the axon itself. This was less pronounced than what we saw in Drosophila, and that's likely because mammals have more glutathione transferase genes, and it's likely that if you were to knock out multiple of these genes, you might get a stronger phenotype. And if you were to knock out all of them, I'm sure it would result in neurodegeneration. So there's a titration issue, which is important when studying glutathione transferases and the impact that they have on both mitochondria and neurons. So just to summarize, knockout of glutathione transferases genes in vitro and in vivo causes increased mitochondrial length. This length was the result of mitochondrial fusion specifically, and was found to be enhanced through the action of mitofusin. We found increased levels of oxidized glutathione alone was sufficient to increase fusion in axons. And these basic mitochondrial findings may be translatable to neurodegenerative diseases such as Alzheimer's disease. And this is now a focus of my group, where redox imbalance have been reported both in post-mortem tissue and in other animal models. So we're looking to see whether manipulating these Gtzt transferases in Alzheimer's models might help with several mitochondrial phenotypes. And so the long mitochondrial phenotype has now been published. So if you would like more information on this molecule and how it impacts mitochondria, please see the paper. My lab now is looking at other mitochondrial candidates and working to characterize those and see how these mutations ultimately impact on mitochondrial dynamics. So that just leaves me to thank the people in my lab, particularly Dr. Daniel Madison, who's doing a lot of work on the mitochondrial projects, Louise Townend, Freya Storer, Freya Sadler, Leo Amadio, Hannah Clark, and Petter Greer, and my collaborator and former boss, Mark Freeman in Oregon Health and Science University, Owen Peters at Cardiff University, Winand, who did some electrophysiology for this project that I didn't have time to show you today, Stefan Zuchner, who does whole genome sequencing analysis so that we can find these mutations, and Hugo Bellin, who has kindly donated a couple of the human transgenic flies for the project. And I would like to thank you for listening, and please do get in touch by email if you want any more information on the mitochondrial projects or other projects in my lab at the moment. So thank you very much and goodbye.